Okay, you guys are all witness, so I just wanted to make it fair. Okay? Okay, Eugene.
And then maybe should you send the email real quick to Sam announcing the result, or would you prefer I write it?
Seung Seungmi, why don't you write
Okay.
in the messenger? Okay.
Okay, thank you so much, Dr. Che.
So anything about the project? What's the plan?
Well, we made the few edits that we made from last live meeting. But we have to kind of make sure it's publishable and see if it works. And then we're going to submit. I just finished my exam yesterday, so this week I'll be working on it.
Okay.
And now I'm not sure where Sam is right now. I think she's in rotations this week.
Okay.
But she and I will communicate and we'll finish it soon. Maybe this week or next week and submit.
Okay.
I'm sorry, but what should I be writing? I don't think I heard all of it.
So Eugene Kim, you just let her know. Like,
Okay.
yeah.
I will message you.
Yeah. Yeah, you can email her back and CC me so that I'm aware. Yeah.
Okay. Okay. Thank you for doing that, Dr. Che.
No problem.
Okay. I'm gonna go to class. I'll
Yeah.
see you later. Thank you so much.
Bye. Okay. Let's see, Dean, do you want to go next?
Sure, yeah. So I was able to meet with Dr. Bhumika this past week. And I believe she went into lab yesterday to get the updated like patient kind of chart information. So I have an updated manuscript to share. So this is still our kind of full length one. I was looking into the GTOCR formatting, and I know that there's a little bit fewer words, but I think we wanted to kind of put together the most comprehensive version first and then kind of cut down later. So I think the main areas, well, I guess, would we be able to read through the whole thing, or should we just jump to the areas where you had some comments from before?
Yeah, just the new area where you.
Okay. So everything in red is addition by Dr. Bhumika about, I think specifically the measurements of the tumor masses from the patient charts, as well as kind of more detail about the timing of the different starts and stops of the different treatments. And then also kind of the intervention or treatment after enicetinib was discontinued. So that's all kind of details provided in red here.
Okay. Side effects, including. We don't use word CT cap. That's just an acronym that people use.
Okay.
Just say CT scan. Okay. Go down.
And then, yeah, I think your comments about the like follow-up measurements and then the the follow-up treatment after discontinuation of enicetinib.
So it says observed from previous CT chest.
Yeah, so we were, I think she mentioned that one of like the collaborators had kind of done the resist part, and we were kind of or she was going to kind of clarify like the wording of that, because that was something that that other person added in. But I'm not, yeah, I think we were still wanting to review that section, or just that one sentence about the resist criteria.
Okay, so my question is, the prior sentence is follow-up CT demonstrated. And then the next sentence is from previous CT.
Previous,
When
yeah,
you say previous, is that previous to follow-up CT? Or is that the CT you're referring to?
yeah, I think I have to check with her on that one. Yeah, I believe it would be previous, referring to previous to this most recent scan mentioned. So 29% change in this CT versus the one that was taken right before that one. But I can clarify that one.
Mm. So there are a few points. Number one, after we started this new drug, anacetinib, the reviewer is gonna ask, did this drug work in the patient?
Yeah,
So the previous doesn't make sense. The CT scan done after how many weeks of anacetinib, right? So, CT scan done post anacetinib showed. Have to be clear. And then because you talked about the fusion and pneumonitis, now you have to talk about the tumor itself. This let's say where is the location and where is the mass, and that mass grew by 29% according to RECIST criteria, something like that.
okay,
But if that's not clear, we should not mention it. If it's unclear, sometimes you measure that something that's not tumor.
Mhm.
'Cause with all these differentiation syndrome going on, it may not be clear. And we don't do RECIST just based on CT chest. So the other question is, was there a disease below diaphragm? I don't remember. So you can only calculate RECIST by looking at the whole body CT, because the sum of the all lesions. So technically you can't say you got RECIST from CT chest, because you
okay,
have to look at the whole body. But I know this CT chest was done because of the effusion, not to do RECIST measurement. However, if we know later on and before the scan, before and after the scan, there was no disease below diaphragm, then we can still use RECIST and say just CT scan, not CT chest.
okay.
You understand what I'm saying?
Yeah, that makes sense. Yeah,
So yeah, special attention to that. And I think to do that, either Bhumi, you, or you can work with Sungmi or Eugene
sure.
to really review the RECIST criteria, and do the measurement. Why don't you work with Eugene O? Because unless you're gonna come to the lab, okay? Because you have to sit down and look and do the measure. I don't know, I don't know if Bumi's gonna do that, because you have to come, literally to here to accurately review the radiology. Okay.
Okay.
So if Bumi or you cannot come in, then just work with Eugene O, okay?
Okay. I can message you. Yeah, I think the comment that Dr. Bumi had was that just based on, because we were trying to identify, I know the comment later on is I think we say towards the end of the discussion that, oh, we, patient reported clinical improvement or something immediately after the start of inicitinib. But I think as she was reviewing like the notes or something, that was a little bit ambiguous. So we didn't know how much of a claim we can make from the improvement as a result of introduction of inicitinib. But I think she's going to double check that one too.
Yeah. So Eugene, after you review it with him,
Mhm.
show me how you guys calculate the resist, okay? So that I can approve.
Sure.
Okay, and then I guess past this section, I updated.
That's
Sorry.
that's red. This one.
Yeah.
So this was the tapering echocardiogram. So ejection fraction is left ventricle. You have to write left ventricle ejection fraction. Okay. Oh, the other thing that's missing is after you use the steroid. It just says after recovery, but that's so short.
Okay.
I think how many weeks after you started steroid, patient's breathing got better, if the effusion resolved, the follow-up CT showed that the effusion is gone, pneumonitis, got better. All those symptoms, what happened to all those symptoms? Just, there's just one word, recovery. Okay.
Yeah. Okay.
How
Okay.
soon did the recovery happen? And the CT findings, did they resolve over time? Did it disappear with the next CT or the next two CTs? And over how much time? No, you have to be very specific.
Yeah.
Okay.
And then into the discussion section, we expanded a little bit more on the different trials. And so I know at least in the approval trial that we reference, I think their claim is that median survival is greater than 9 months for patients with refractory relapsed AML who are unsuccessful with previous treatments. And they compare that to, I guess, recent to that time, a different trial, which found only a 3-month median survival. So that was one point that we added in. Yeah, so for that one trial, but I think we can also provide more explanation of that too. But I guess again, their central claim is that there is good tolerance, induction of response, and then a much improved median survival compared to what's previously been given for these patients.
That's okay.
And then, yeah, I think the other studies, we just kind of added a little bit more supporting detail and references. I know Dr. Bumi still has to, we have to correct some, but I think we have the relevant references for all the claims that we're making, just not necessarily in complete numerical order yet.
Wait. You talked about overexpression. What about mutation?
For this reference.
Because the sentence starts with, apart from mutant IDH2, But what is, can you go up?
Yeah.
So if you go to see bioportal, you can look at the overexpression, but you can also look at the mutation
Mhm.
of IDH2 in lung cancer database. But it's never mentioned. It just says apart from.
Okay.
So you have to mention the prevalence of IDH2 mutation in non-small cell lung cancer. And what percentage of those mutations are really thought to be functional? And Grace and Eileen can tell you what's functional. Anyway, so those are the questions you have to answer.
Mhm.
Okay.
Yeah.
Okay. Okay. Go down. So clinical improvement. So did you find in the chart?
Yeah, so that I brought that up to her, and I think from what I understood, the notes were like, I think she recalled that there was a note or like a claim made by the patient somewhere where there was, self-reported improvement. But I think she wanted to go back and double check that, because if we do want to make this claim, then we would add that mention also to the results section,
Yeah,
that there's kind of self-reported improvement.
yeah. So yeah, add that. It's like I I think I remember patient saying she felt better. But that wasn't really supported by the next CT scan because of the differentiation syndrome.
Yeah,
But you never know.
yeah. So maybe like the time frame, like within immediate, not immediate, but like with sooner, much before the
First couple weeks,
interesting.
something like that,
Yeah,
and then followed.
yeah.
So what was the onset of this syndrome? Was it 2 weeks? What was it?
So it was 53 days, or about, I think that's a little over 7 weeks.
Okay.
And so, yeah, so I think we said that that time frame is much longer than other therapies, but
Okay.
and then like kind of lines up with existing data.
Yeah.
Yeah.
Okay. So 48 days, 11 days. So what do you mean by other therapies?
Uh,
No, go the reference
yeah,
12.
yeah. To double check that one.
So you have to say other therapies such as, what did that reference have? Atra?
Um, just double check. Yeah, compared to ATRA and ETO. And then we also add that claim here. So the occurrence and then also the duration before occurrence of.
Yeah. Such as other therapies, including Atra and ETO.
And then I think I meant to also order this so that we say that enicetinib results in delayed occurrence, but also less occurrence in the first place. And then yeah, so I think I'll make those claims right next to each other so that that's more clear.
Okay. Good.
Okay. But so yeah, so I'm going to share with Dr. Blooming again, and then yeah, we will correct that resist criteria. And then I'm also talking to Dr. Liesel and others about just proofreading once we have that more final version.
Yeah, yeah. You're almost there. Almost there. This is not
Yeah.
easy. You know, case report, you have to know ins and out, the mechanism and everything about the patient. So, I mean, it's not easy on your level. However, just learning to work with doctors and understand
Yeah.
how to write patient-related findings and mechanism of action. So I think it's a good learning experience for you. Yeah.
Yeah. And I can also follow up with Dr. Elena and Dr. Pedro, because I know that we reached out to oh yeah, we at least heard back from Dr. Elena, but I'll make sure to follow up with her too.
Yeah, we gotta finish that. I don't know why it's taking so long. Um, so I know they're waiting for statistician, but other than those numbers, we have to be done with discussion and everything. So don't just wait for the, just
Yeah.
work on the manuscript, introduction, discussion. There's many things to finish. So
Yeah.
yeah. And then one of my residents what's his name? Dan, was it Dan or
Oh, Ben
Ben?
Bryant.
Benjamin.
Yeah.
Ben. He wanted to work on it as well, so you guys can work together to get it done.
Yeah. Yeah. I've gotten in touch with him, and yeah, I think I'll make sure I keep following up with
Okay.
her, and then also with him in.
All right.
All right. Thank you, Dr. Shea.
Thank you. Very good. Okay, Eileen. Updates.
We haven't heard back from the Wix thing yet. And this time Dr. Liam was saying that I should just be more like assertive and like call for a manager. I don't know how I'm gonna do that, but I guess like the next time I chat, I'll do it again. But I just wasn't able to do it yesterday. I made a list of questions that I want to ask Mr. Curie for his interview.
Okay.
And then after talking with Dr. Liam, I think I want to try to interview him next Monday. So I actually just contacted him this morning. I
Okay.
sent him a text message asking, or like I brought up like who I am and like what I'm doing, and then
Yeah.
asked
Great.
about next Monday afternoon when I come
Do you
in.
have his cell phone number?
Yeah.
Okay, good. Who are you
And
going with?
Dr. Liam.
Okay, Dr. Liam. Okay.
Yeah.
Great. This would be so yeah. You heard about two versions, right? One for pacemakers, the other for grapevine ministry. Did you hear that?
Oh no.
So we're gonna have different questions all mixed, but basically we're gonna, one is more like faith-based. We'll go to Grapevine TV and like doctor-patient relationship based questions and answers will go to Pacemakers TV.
Which one am I doing? Am I doing?
Well, it's going to be the same interview.
Oh, okay.
You just ask same thing, but then when you edit it, the faith-based ones will be, pulled together.
Oh, okay. I know that like for the one that we did for you, it was 20 questions. So am I still doing 20 regular questions and 20 faith-based questions, or?
No, I think you could do, I think ideally we're looking into 10-minute clip
Uh-huh.
and for each. So it's 20-minute total. For 10-minute, I don't know. 10-minute, is that 5 to 10 questions usually, maybe?
I think 10 is a good number too. So
Yeah.
And it could be more in-depth questions so that he takes more time to respond.
So
I did make 20 just in case.
yeah, I think it's better to email him or text him the questions like a day before.
Yeah, I said I would do that.
Yeah, so that he can kind of get prepared.
Yeah.
And remember, it's better to do walking interview, but he might get shorter breath because he might be still a little bit deconditioned.
Uh-huh.
So maybe like walk really slow, but
Right. We could also do sitting, or did we want the walking format? Would that
I like the walking format, but it's
okay?
up to him. He might get,
Yeah, for sure.
but at least first couple minutes, if you can walk, and then maybe you can kind of start by walking and then walk to the chair and then sit or some, maybe I just want to have that walking setting, if not the whole 100%. Good. Yeah, yeah. And then
Okay.
the questions you can just run it by um Dr. Liam.
Yeah.
Good. Very good. And then Shauna can help, edit and
Editing.
clips back and forth and
Yeah.
help you get the thumbnails or things like that. So get her help.
I haven't made YouTube videos before, so I might need some help on that.
Yeah. I think Liam and Seungmi can help you too. Seungmi in the lab. Okay.
Oh, okay. And then
Yeah.
also at the undergrad meeting, I think Grace mentioned for the VC project,
Yeah.
I think the due date is apparently this week.
Yeah.
From what I heard. And I just wanted to I don't know if I have the bandwidth to do it this week just because a lot of my final projects and exams are this week.
Yeah.
So I don't know how much of a help I can be until Friday.
Right.
So I'm willing to back out for that submission because I don't know how much of a help I can be, but I'm willing to help on Friday, but I think that would be too late. So I wanted to let you
Yeah.
know.
So maybe not for the abstract, but for the
Yeah.
actual project, you can do
Yeah.
more. Maybe. Yeah. Anything else?
That's it.
Okay. Then about that project, let's hear from Grace what the plan is.
Dr. Che, for some reason I thought the deadline was the 15th of March and not this week. So I'm in full panic mode because I don't know if I can finish on time, but I'll share what I've done so far.
Sure.
If in the chance that we can't submit by this Friday, is there a different conference we can go for?
I think this is our best bet. Otherwise, we wait
Okay.
all year, which doesn't make sense. This is
Alright.
a lung cancer dedicated conference. And we have a lung cancer experimental. So I think Eugene, you're done with protocol abstract, right? Do you have anything else to submit?
No, I don't think so.
Yeah, so Eugene Kim can work full day until, is it Friday? Is it Friday?
Deadline?
Yeah, the deadline is Friday.
Yeah. So I mean, it's just,
I actually I think I can crank out most of the tool things because I'll show you what I've done soon, but what I might need help with is whatever R analysis is required. And I know Dr. Eugene is a little, at least more than me, familiar with R.
yes.
So that might be a little helpful.
Yeah, I
could
think
get help
with
on there.
Yeah, help from Eugene, you can finish it.
Yeah. I'll show you what I've done so far.
Sure.
My
Yeah.
goodness. Can you see my screen? Okay, so for I was able to go through the CBio portal and get the first 10 if possible for each gene that you mentioned, Dr. J. And I decided to keep the ones with at least 2 databases within CBio Portal. So CBio Portal includes 4 different databases. So
Alright,
if
see, but
there were, yeah, if there were at least 2, I just included it, and I went by highest frequency, the variants with the highest frequencies. And so this PIC3CA was fine, but mTOR only had 9. And then the FGFR, I couldn't find any for FGFR1. Like if you see here,
Hmm.
none of the missense mutations, these were all the missense mutations that showed up for FGFR1. None of them had conclusive data from more than 2 databases.
So how do you know it's more than 2?
So like if you look at the annotation column here,
Yeah.
like if it has like, that's OncoKB, that's Civic, that's My Cancer Genome, that's Cancer Hotspots.
Yeah.
Like that gray is inconclusive. That blue means that OncoKB says it's pathogenic. This means that these 2 say they're pathogenic, but these are fusion mutations. All
Oh,
the missense ones, like there's nothing for FGFR1. I
interesting.
could only find it for FGFR2, and that was only like 4 missense variants that I could find.
Got it. Makes perfect.
Yeah, IDH1 was like about the same. P10 was good. MAP2K,
They're all valuable observation, Grace. So
yeah,
remember this word, just put it down somewhere that, you know, in FGFR one, there were, you know, the pathogenic ones were all fusions. So these are
well, okay, all
right.
right.
Yeah. And none of the missense mutations were, you know, found to be pathogenic. It is
Yeah,
good.
it just didn't have data. Like, I don't know if you could say they're not pathogenic because you don't know if they're benign.
Oh, I'm saying none of the missense mutations in the TCGA database or GBioPortal database were thought to be pathogenic, or
Okay,
that's what I meant.
all
Yeah.
right.
Okay.
For stuff like, oh, it was the same case for MAP2K1.
Mhm.
Oh, IDH2, I could only find it for all IDH1 and not IDH2.
Good.
MAP2K1, but not MAP2K2. For
Wait,
things like,
wait. This
yeah,
so IDH2. So
yeah,
Dean Tan just talked about IDH2 inhibitor used for non-small cell lung cancer because patient had IDH2 mutation.
yeah.
But that mutation, at least from the CBio portal, we have to look at what it is, may not be pathogenic. Therefore, IDH2 inhibitor might not have worked. So so
Yeah.
oh, so that's a great. So Eugene O, remember. So talk to Dean Tan. I know he's left Zoom, but and talk to him about the what is the exact variant? It's IDH2. What is it? And then work with Grace to see in CBio portal, is it reported? And if it is reported, is it not functional as she just told us? I think writing that is a vital piece in the discussion. So that's
Would
what's
that be for the abstract too?
for. Oh no, this is for discussion.
Just for, okay.
Yeah. We don't need it. But Eugene, for that case report discussion, we need that.
Oh, okay.
So talk to Dean, okay?
Okay.
A question I had was first for the ERK pathway, you asked me to look into MAPK1 and MAPK3. MAPK1 had 1 variant and MAPK3 had none. Should I include this or not?
Let's include.
Include it. Okay.
You know what? Um, I think
Yes.
I know you went with at least two, but there are
Yeah.
ones with at least one. I mean, we have to be consistent.
Well,
You already did with at least two, right?
yeah, I just did at least two. Okay, but this one I just listed even though there were just one because there was nothing otherwise, and I
Right.
didn't know what, like I just put it as backup, but I think it's better to just go with things with at least. Oh, but Burka Mutations only had one too.
Right.
So I was very confused.
Right. So I think we should be consistent and make it at least one. But it doesn't mean you have to go back and change. You know, if there were more than, you know, at least two, those were prioritized, you can say in the method or something. So that you can keep what you have, but for the ones that didn't have, you know, two, at least two, then we included one. Also, something like that. In the methodology, you have to write out how we screen for these variants.
Is that fine? I'm just wondering if that's too complicated.
I mean, that's not ideal. I think ideally, I would just say in the order of frequency, and the criteria is at least one. Because, you know, the whole idea is we're assuming that because at least one identified as pathogenic, there's a chance that could be pathogenic. And then what's the concordance for those? You know what I mean? Like, you know,
Yeah, but if it's just one database, okay.
so it's not like we're gonna say this is gold standard, and then what other, you know, platforms are doing. And it's mainly on KB if you look at CBioportal data, right?
Yeah.
Yeah. So I think it doesn't matter in the end.
So go back and do the one database one.
Uh, so I would. So you prioritize, just say you prioritized ones with at least two.
Mm-hmm.
But then for the ones that did not have two, you did include ones that had only one.
Okay. For example, for like mTOR, if I had like 9 with at least 2, but I was missing a 10th one, should I add in another one or just leave it at 9?
Add another one to make it 10, because I want to
Okay.
be consistent. Yeah.
Okay.
Yeah, just one more.
All right, I'll go back and do that. Oh gosh. I started writing the shell of the abstract. I also didn't know how and who to include in what
So
orders.
So it has to be whoever did. So I want to give Pedro, I think he's awfully busy. I wanted Pedro to sort of take the lead, but I think he's very busy. But I know he met with you once, so right?
Yeah, yeah.
So I want him to be the last author,
The last.
and just take out my name because I have enough of variant classification. First, I don't need any more. I'll just be the mentor behind the scene.
Okay.
And as a, because you know who did what, like in terms of like, you know, work distribution and so forth. So
The problem is I haven't been very good with reaching out, so this is all last minute, so I haven't like assigned people things, which is the problem. But I know they'll all be contributing for like the bigger project in general. Like just for the abstract, I've just been cranking things out. But I don't want to like take people out because of my, you know, like.
So let's assume that all these people will actually contribute. So Levi,
Yeah.
Lina, I know Lisa, like she's working, right? So like I don't think I don't know if she has bandwidth, right? So still leave them, and maybe you can ask them. So Eileen told us she doesn't have time. You can ask Iser, Lina, but we know Eugene O can help. So you can add Eugene O somewhere. Yeah, there, Eugene O. And then just you and Eugene can divide and conquer, and see if Lina can help out with some. Yeah, and because we are, I think once you have the list, then the rest of it is just run the R, right?
Well, run it through the tools and then run the R.
Yeah,
And
running
then
through the tools. Yeah.
I've started on the tool stuff too. It's just, yeah.
So how about, because this is being recorded, how about you sort of think of this as a tutorial, show like just couple of gene variants, how you do the calculation, and then
How the calculation. Oh, okay.
yeah, Eileen, Lina, Eugenio can kind of, and Isa as well.
Yeah, that would be
There is a recorded video that Emma made that
Oh,
is
Emma.
very comprehensive of how each of the tools work.
Okay.
Um,
Well, why don't you
so
just quickly do just
I
one variant? Just yeah.
okay for all tools. Okay, I can try. So basically, I have the sheet that organizes each variant with like the input codes for each tool. And then I'm going through and doing all the Condel ones because that is giving me the code for mutation taster and other stuff. And so this code is, since this isn't PIK3CA gene, the P42336 is basically something that indicates that it's within PIK3CA. The 420 is the location of the variant, and then the R is what it's changed to, the last letter. And then basically you go to,
Oh, wait.
there's a,
Before you do that,
yeah.
can you show them how you got all these blanks filled in by going to their platforms?
Oh, these are the inputs that you have to put into the platforms to get the results. So these are like the codes that you enter into the
Show us how you got these.
yeah. So basically the
That's the variant
P,
you got from C
well,
Bio C, right? That's
yeah, so the variants I got from CBioPortal based on their frequencies and
yeah.
the top 10 frequencies.
Yeah, show
And
them.
then,
Yeah, from C420R. How did you get that?
oh, like in CBioPortal
Yeah,
itself.
in C Bioport itself. I just want people to know.
Okay, so I went to lung. I didn't select this one, and I didn't select the small cell lung cancer because we're focusing on non-small cell. And then query by gene, and then this would be PIK3CA. And you could also include like multiple genes at once, but for this purpose, I'll just do PIK3CA. Once this pops up, you go to mutations. And then I arrange this by allele frequency so that the highest frequency one pops up first. And then you could see there it has at more than 2 databases. And if they're colored, that means they're pathogenic. It means the tools predict they're pathogenic. You could check by looking at here, it says oncogenic, and like this one, you have to like click to get in and stuff like that. But anyway, I would take the C420R. This is in this sheet, but I would put the C420R, the frequency, and then for OncoKV, Civic, and cancer hotspots, it said it was pathogenic. So I put a 1, 1, and 1. And then I basically went through the whole list, got the first 10 of the most frequent ones with at least 2 databases. There were some repeats, like E542K happens like multiple times, so you just have to be careful. It's just because it's included in multiple studies. But once that was organized and I got a list of all variants for each gene, I came up with the tool inputs for each of the variants too. And these are the 6 in silico tools that are going to be tested for this study. And each of them require a different input to identify each variant. So polyphen-2 and Condell require the PIK3CA gene identifier, the position, and then polyphen-2 has the starting amino acid and the new amino acid that is changed.
So where did you
And
get that P number from the CBio portal?
So that wasn't from CBio portal. That was, I went to,
Just
I
the
would look it up in like NCBI,
Oh,
look up PIK3CA, and then get the code
yeah, show people.
for, I think
Three.
it was,
Gene. Yep.
and then it would have to be under Homo sapiens.
Yep.
Maybe I did not get it.
Yep.
I might have gotten it from a different website, but it's like the same idea. You look it up and make sure it's within Homo sapiens. P423. Yeah. Well, this one is, yeah.
What website is this?
This is just NCBI, but I think I did it in, ooh, where did I do it? I think I looked it up in a different database. Yeah, but this is NCBI and it should have, oh, I think I looked it up in UnitPro. Oh yeah, I looked it up in UnitPro. And then if it was PIK3CA Homo sapiens, then the entry number here would be the identifier.
I see.
And you just need to make sure it's not like mouse or like some other gene.
Right. Yeah. So you got that one, and then.
So I got that from there, and then the 420CR or
Yeah.
the 420R is just from the variant information.
Okay.
And then align GVGD is just like the name of the variant, so that's pretty intuitive. And then the mutation taster IDs come when you run it through Condell. And I could show you Condell. The Condell website was down yesterday for some reason when I tried to do it, so I panicked thinking we can't use Condell. So I tried doing everything one by one, but it wasn't accurate. But now it's working again, so it's fine. So for example, if I go back to the PIK3CA example, I can run a batch query for Condell. So I'll take the Condell codes, and then this is in query Condell. You put in all the mutations here, and then you could just like set a name for yourself so it's easier to remember which one it is. And then this one, you just download the file, and then I'll share my screen again. And then it gives it to you in this like Excel sheet format. And basically you take this information, you can transfer it to your Google Sheet. For example, like I'll paste it down here again. And then you get the mutation taster code by putting the chromosome number, colon, the start position, and then T arrow C. But I'll go through and do all of those so people don't have to do that. And then that's what you enter into mutation taster. So it's better to do Condell first in order to get the input for mutation taster.
Got it.
But anyway, like this is 420 CR and 420 CR. And then basically I kept the data from here. And this is saying Condell predicts it to be neutral rather than pathogenic, which is in conflict with the databases. But I would put that data here. And then there are some like EK that like repeat. So you just need to like go through and see if the codes are unique and just clean up the data. And then that's how Condell is run. Should I explain every other tool too, or?
Yes, I want you to just at least show them.
Okay. I have not run every tool yet, so I might not be familiar myself.
Okay.
Polyfen 2 should yield this one. Oh, okay. This one is the one with the FASTA file. Okay. I'm gonna do this. Okay. So I'm gonna go back to this one. Polyfen 2 codes are here. So I would paste that batch query. And then this one, you have to upload. Oh, I don't know if you have to add this. I don't know if you need an email. I'll try without it. And then you have to upload the FASTA file. I mean, it says it's optional, but Emma did, so I just did too. And the FASTA files I'm gonna collect in here. I have the one for PIC3CA. So what people can do is download it from here. And then when they do this, they could choose file. And then, I don't know where to download it. My screen is frozen. But you would, okay, but you would attach it there. And then, yeah, my screen is dead.
So why do you need FASTA file there?
I'm not sure. I also don't know how to get the actual like FASTA files themselves, so that's something I need to ask Emma.
But why do you, I don't know why you need it. It's just a text file with the sequence.
Yeah. It does say optional, but I know some other tools actually require it too. Oh my goodness. I'm gonna quit my browser real quick.
Okay.
Oh. I guess just trying to upload the file just freezes my entire browser for me. So I don't know if I can show that right now.
Skip Polyfen then.
Yeah. But I mean, Emma's video is pretty comprehensive and it should help people. Okay. Okay, back to PIC3CA. We'll do align GVGT. Interesting. I think- let's see. Oh. Oh. Hmm. Oh, Dr. Che, I don't remember how to do this.
Mm.
Mm,
But we gotta write picture CA somewhere. Mm.
yeah. Because I personally only did one of the tools last time. I did CAD,
Mm.
and I'm still learning. I mean, I did watch the video yesterday, but I didn't retain everything.
Mm.
But I remember Align GBGD was pretty simple. I just need to go back and make sure I have all the inputs I have. But you basically paste it and it should give you everything.
Yeah,
Um,
just that's
and then GBGD just spits out an input that says like class equals something and a number. And if that class is greater than 35, that means it's interpreted as pathogenic. And then mutation taster has the inputs that we got from before. Oh, I think mutation taster was the very annoying one where you have to do it one by one. I'm pretty sure. Yeah, you have to do everything one by one. So that's like a lot of work. You can't do it batch query. So you would have to like, I don't know where to put it. Transcript. Mm,
Yeah, Dr. Say, I think we can figure out all the other programs by seeing the video made by Emma.
yeah, I think that would be better. I haven't done everything, Dr. J.
Okay, let's do that then. Show us
Yeah.
the calculation then. Let's assume that you have
The calculation.
the concordance.
I haven't. Okay, I have to look into that too, Dr. J.
Okay.
Yeah, but like once all these tools are done, we just have to gather it together, and then it's plug and chug from there. Compare it from between the database.
So let's so today's Tuesday. So in terms of finalizing the variants, what do you have to do, Grace?
Well, now I have to go back and add the one database ones, I guess.
And that's not a lot, right? That's just a few,
Yeah,
or
it should be a few. Yeah.
yeah, it should be a few just to get to 10, right?
Mm-hmm.
So
I
that
can finish that by tonight, and then I'll also have all the tool inputs ready by tonight. And then if we could just split
Grace.
up, yeah.
I can help running through like one or two of the tools like tomorrow night, if that's okay with you. I just don't know if that's like okay with your timeline.
No, that would be great. That would actually be great.
Tomorrow,
Unless like I get
like,
most of them.
Okay, yeah. You should assign us like a tool each for now, and then or like assign me 2 tools or something, and then I'll create those out as well.
And then like I have the sheets where they have to be collected. So you just have to copy paste it in there. And then I'll just clean it up. And then the analysis Dr. Eugene can help with.
Yeah, so Eugene, you have to help with the R portion where so they'll get which one is pathogenic per with each platform. So we're the question is what's the concordance between the platforms?
Or is it like basically, oh, can you hear me?
Yeah.
I'm sorry.
Oh, so sorry for the interruption. But like, is it like basic? Is it like ANOVA comparison between the gene group or? But I can figure it out later. Like,
So there's a manuscript written by Eugene Kim,
oh, I'll look at it.
and he shared with us last lab meeting too. Or Eugene just, well, Grace, can you send Eugene that manuscript?
Yeah, I'll find it and
And then
send it. I'll also send the R file that Emma sent me. Maybe that will help.
That
Yeah,
would be great. That, yeah.
video
Yeah.
and video link.
Okay.
So
Thank you.
Eugene, today you figure out the video and how to run the R. And Grace, you finalize the selection of the variants plus the annotation. And tomorrow, if you can, and the Eugeno can help too. Tomorrow, Eugeno, Eileen, Lena, Grace, you can work on what's pathogenic, what's not pathogenic, that. So that's Wednesday. And Thursday, Eugeno, you can do all the calculation according you running the R. And then once you Grace, what you're gonna do on Thursday is just write the abstract.
Oh, can you take a look at the intro methods for the abstract that I wrote so far? Just
Yeah.
because I probably won't see you in a lab meeting.
Right. And then we can plug in the
yeah.
number once we get it, right? Yeah.
I just have this left. Well, this is from Emma's. This part I could write without the data, so I just wrote it for now.
Yeah.
I'll tweak the methods for the one database thing, but otherwise, can you just go through it very quick?
Sure. Let's start with the title. Good job. This is what I want. Yeah. This is a good practice. Don't wait till you get the data. Write it all out, and then you'll just, put in the blanks, the numbers. Valuation of in silico tools for variant classification in missense variants. With non-small cell lung cancer with potentially actionable targets. I don't like the width in missense variants of that are. So these means that what missense variants are potential actionable targets. So how do you?
I think you meant like potentially actionable target genes, right?
Yeah.
In missense variants of potentially actionable non-small cell lung cancer target genes.
No. So the targets are variants. Missense variants are each targets. The genes may contain non-targets and targets. So it's not the gene that is target, it's the variants that's target. So maybe not variant classification. In silico tools, to determine actionability.
Oh,
To determine actionability in missense variance. Of
yeah,
missense variance.
that was taken mostly from like previous,
I know.
like for variant classification in clinically actionable and missense variance.
Potentially actionable. Clinically. Well,
Yeah,
but wait. Let's see if this makes sense. In silico tools to determine actionability of missense variance. Or potential actionability. Put potential actionability. Determine potential actionability of missense variance with experimental therapies. With experimental therapies. Yeah, and just actionability of missense variance. That makes sense because we're not doing standard care. We're not doing the FDA approved targets. We're looking at potential targets that can be used to treat lung cancer using non-standard of care. Missense
It-
variance.
the title can be 125 characters, I think, including spaces, and we're exactly at 125.
Oh. Experiment therapies. Do you like for lung cancer or in lung cancer?
With experimental therapies I think 4 makes more sense, but I'm not completely sure.
Let's leave it like this for now.
Okay.
Or introduction last page, that's in silico tools to predict the actionability of potential, well, pathogenicity of potentially. Okay, leave it, it's okay. No, I like the introduction method. We have 15 genes.
Well, do we count like AKT 1, 2, 3 as separate ones?
Yeah,
Right?
we're gonna, yeah,
Can I write it like this, or should I like spell
write it.
out AKT?
You can write it.
Okay.
Is it 15 then?
Yeah.
Okay. So you at least two or more. So we have to say at least one or more just to,
In a sense, with at least two or more we're prioritize.
yeah.
Um, we're included.
And then you have to say, you know, what would you say classification one? So the classification is of pathogenic,
Yeah.
because it's not clear, at least concordant classification of pathogenicity. Yeah. Yeah. That's fine. We then,
Is this necessary?
no, as necessary is fine.
Okay.
So in six widely used six in silico tools, to say, yeah, that's fine.
Okay. And then I'll crank this out once we have data.
Yeah, sounds good. Sounds good.
Okay.
All right, we can do this, Grace.
Yeah.
Okay,
Um,
just work closely with Eileen, Lena, and Eugene.
okay. Yeah, that's all for me.
Yeah, I think, yeah, we got the final list almost, and then it's just getting each platform's pathogenicity, yes or no, and then just do the calculation for that sensitivity, specificity, yeah. All right, so this is abstract. That's why I didn't want to do the full thing and said let's cap at 10. But once we submit the abstract, I think we should go back and say we don't want to cap at 10, but we want to look at all the variants. And each variant, the list you created, Grace, will be all supplemental figures, supplemental tables for each gene, so that when people look at our article, they go to a website and download supplemental figures, and they don't have to search all, like they can use our article to screen for, hey, is this really like a potentially a good target? They can use our article as a reference paper to look at potential good targets for lung cancer. So yeah, these all like per gene generated tables could be all good supplemental tables that we can actually upload. And everything else is going to be really similar to what Eugene Kim wrote. Just the only difference would be FDA approved versus FDA non-approved. So it's a simple concept. All right, any questions, Grace?
No, I'm good. Thank you.
Anything else you've been working on?
No, this has taken up my whole time.
Okay. So you are taking the lead, so I want you to, you can be the first author for this. And I don't know if you have fun to present this. So look for any student funds, and I can.
Oh, if this happens to be accepted or anything, I can't go because the conference is in my first week of medical school.
Oh really?
Yeah,
First week.
yeah.
Okay. Then you would designate a presenter who has time to go if someone else is going. Like Eileen, if you want to go, you can be the presenter. Or Eugene O, or Isra Kim, whoever. Okay. Okay. Very good. Thank you, Grace.
Thank you.
Let's go to Sungmi.
If it's okay with you, like, can I go first?
Oh sure.
Okay. So for me, like, the only thing I want to present is the abstract for like the protocol that we're working on. But like as you said, as you mentioned yesterday, I'm not sure like the word long require us to put like clinical trial number. So just give me a sec to. So I'm trying to figure out that, and can you see the screen?
Mhm.
Okay, so this is the abstract page for clinical trial,
Mhm.
and it says like it seems like it does need clinical trial number, but I think so. Do you think it's worth to ask them like separately for does it really necessary, or because I can move to the other section, but yeah, but it really seems like it needs the number, right?
Yeah, it needs the number, so
Okay.
don't bother then.
Yeah, okay, then for this time, I think I cannot submit my protocol. And then, but at least I can show you the revised version of the protocol itself.
Yeah.
Okay. Sorry. So I highlighted the revised portion as blue. So for the first part, like as you mentioned, like I revised this portion just like the word that you used in your presentation, like in the tumor board.
Wait. I think
Mm-hmm.
clinical trials in progress. What about the general
Abstract.
abstract? Can you still, is there a limit?
For general abstract, I think that only, yeah, I don't do it by myself, but I expect them just require us just the abstract, not the clinical trial number. But because there is a classification of clinical trial, that's why I choose this one.
Can you go to general abstract? And I want to look at the categories.
Oh, okay. So this is still for clinical trial. And so if you see this page, you can choose the kind of abstract you want to submit. So the previously I went like this, this one, but if you go regular, then I think you can just, maybe I can just enter the title. Yeah, just, I'll just,
Anything, yeah.
yeah. And then this is like the same with the clinical trial version. So maybe we can choose. Actually, I didn't choose anything like because I was not able to choose the appropriate one because we're dealing with metastatic, not metastatic, but
Mm-mm-mm.
do you have any preference?
Mm.
It's a little bit restricted, I think.
Well, we have to check surgery.
Okay, the early,
Locally
locally,
advanced, yeah.
yeah, okay. I think there's nothing we can put it in here and then, oh, did not fill in, oh, okay. Yeah, after that, like, I think it doesn't require any trial number, like something like that.
Mm-mm-mm-mm.
Yeah, maybe we can just try this version, but I'm
Yeah.
not sure.
You
Oh,
can
you
try.
wanna? Okay. Yeah, then I can show you the abstract that I'm working on. So this is
Yeah, show me.
okay.
Yeah, it's just submit, see what happens.
Okay. Can you see this version? The Word file?
No?
Oh. Yeah. I'm sorry. Just give me a sec. Uh, Oh, can you see it now?
Yeah.
So I divided the whole abstract into rational and objective and methods because we've not yet started it. So we don't have any results or something else.
Mhm.
But I found out that the rational portion and the methods is pretty large, like almost taking up half of the paragraph, half of the whole abstract. So I'm not sure if it's right or wrong. And for the title, there's a restriction, like 125 characters. So I ended up like revising it as trial for what do you think about it? Like I think this would be appropriate for the length restriction.
Yeah. That's fine.
Okay. And we can just put any like other one's name if they can revise it.
One of the absolutes. Reported introducing a subset who underwent lung transplant, subset, BAC, lipidic.
I'm sorry, maybe I can just,
Yeah, that's fine.
okay.
Our study, what is that?
Our study is mentioned here. Our study group, in our study group. Yeah, he underwent.
Can you change the setting, like the one word doesn't get separated? Is that there option to do that?
Oh, do you mean the section of each abstract?
Yeah, the paragraph, can you make sure the words are not separated like a group?
Oh, okay.
There's a way to do it, right? No, I'm saying
Mhm.
just like the word group, group. Can you,
Oh, yeah, got it. So you don't want like this. So
right? I want,
yeah, let me figure it out later. Yeah, oh yeah, what's
there's a way
happening?
to do it. There's,
Okay. Yeah, I'm sorry for like this kind of thing, but yeah, I'll make sure to revise it.
yeah. So this, the case,
Mm-hmm.
I don't think
It's
we
not
can write
published
it
yet.
here because we are submitting
Oh,
separate case report.
oh yeah, that makes sense. Yeah,
Yeah,
then just like
let's
erase all the
take it
part.
out. Yeah.
Yeah.
Rationale, objective, those we aim. Prognosis and molecular. Now, I wouldn't say prognosis. I would say outcome.
Okay. Okay.
Clinical outcome. That's the best option for survival.
Um, Actually, I refer to it from the Ankit.
Yeah.
um, or for whom the long term will be performed as.
So I would have subset of it long. So I'll emphasize ones without, metastasis to lymph nodes or
Oh, okay. So
organs.
yeah, without or I'll just write it abbreviation for now. Node or any distant metastasis. Or involvement.
I'd say lymph node involvement or distant metastasis.
Mm-hmm.
For whom long transplant will be performed as the best option for survival. I don't like the best option for survival.
Mm-hmm. Yeah, it sounds a little bit radical. So performed as the
For whom
or
long
we
transplant,
can just erase all the part.
yeah. And I think we're just gonna say double.
Oh yeah, for sure.
So
Double.
for double everything, right? Double, even the title.
Okay.
Yeah, I think it's good.
Okay. So yeah, I think so. there is a word restriction as 500 letters, words, and I'm pretty sure this will be 400 or something. So if it's okay with you, I'll just go with it. But I think, yeah, we can add something else, but yeah. Can I just finalize it as it is?
Yeah.
Okay.
So because this is going to be the protocol, go
Mhm.
up, go up. In the abstract, we
Mhm.
don't, we can. I think you should put yourself as a presenter, but because this will be a clinical trial, I don't think you can go first.
Oh,
I'll
okay.
go first. You put your
Mhm.
name second, but you can still say you are the presenter,
Mhm.
and you can still say you are the equal contribution first author for the abstract.
Okay.
And
Yeah.
then we have to put all the surgeon's name because this will be a surgeon's protocol. So Samuel Kim, right? Samuel Kim and
Uh-huh.
Ankit Bharat.
Yeah.
And then we'll let Ankit add any surgeon who will be involved in this trial.
Mhm. Do you think I need to just send this out to the surgeons, via you?
Yeah, I will send it out. So
Okay.
once you have the final version,
Mhm.
send it to me. I will send out to surgeons CCing you.
Okay.
Yeah, after your name, maybe add Iryong's name
Okay.
because he's gonna be involved in more details, and have him proofread and change anything so that he does something.
Okay.
But yeah.
And what about the protocol itself? Do you need it as soon as possible, or?
Yeah.
Oh.
Well, protocol, do it after you submit this. And what happened to the case report?
So for Curie, right?
Yeah.
Isol told me like she revised the abstract, but we're not working on the case report yet. So I'm sorry for the delay, but you wanna
To send that Albert Curie case report to the surgeons.
oh, you have to. Okay.
I have to send to it today so that they have at least 3 days to look at it
Mm-hmm.
that we can submit on Friday. I cannot send it on the day of deadline.
Right. And you cannot send just protocol itself. You have to send it with a case report.
So this abstract
Mm-hmm.
is not protocol. This is just
Oh yeah, this is
this
abstract.
is called just the introduction of clinical trial.
Right.
So this one I have to send to the surgeons today.
Mm-hmm.
Case report I have to send to the surgeons today so that they have time to look at it. Usually you have to give them a week.
Okay.
So we're already
Okay.
late.
Oh, you mean that so we need to at least finalize our draft for case report by today, right?
Right. It was almost ready 2 weeks ago. I don't know why it's taking.
Oh, okay.
So case report. So think of this as a collaboration with surgery. So
Mhm.
we can, we we have to. It's their specialty. It's surgery. Okay. So
Right.
they have to have enough time to revise, go back and forth.
Yeah. Making an input.
So we cannot submit it. If we were only giving them a day,
Oh, okay.
or two, even we have to give them a week. That's the courtesy.
That makes sense. Then I'll focus on the case report itself, like receiving help from Isol so that we can finalize the draft. And
So it has to be sent today.
okay.
Okay.
Yeah.
And this has to be sent together. So I'm gonna, We will send it out together.
Yeah, okay. The case report and the this kind of abstract, right?
Yeah, trial abstract.
Okay.
And once we send it out, then we finish the protocol, the action.
Oh, okay.
And then we send out the protocol and we get,
Got it. I got it.
that's the sequence.
Okay. Okay. Yeah, I'm not sure I can do it, but let me just give it a try.
Yeah, yeah. I don't know why case report got lost. Because I thought that was almost done when Lisa presented. I gave her comments and yeah.
Oh, have you given her like the comments on case report itself, not on abstract?
No, I'm saying the case report abstract,
Oh, yeah.
not the full case report.
Oh, I'm sorry. I got it wrong. Okay, case
Yeah.
report
I
abstract.
gave all my comments about the case report abstract.
Yeah, for sure. Yeah, I think in that case, we can definitely finish it and then
And
send
it's just a
send it.
couple paragraph. It's
Yeah, right.
so I was hoping to send that out
Today.
at least a week ago, but we're still
We
so
run
let's send
behind.
it out
Okay.
today. So
Yeah.
not the full case report,
Okay,
but the
abstract.
but the full case report has to be sent out soon as well. But
Okay,
yeah,
but not as urgent as an
yeah, not
abstract.
as urgent. Yeah. Okay.
Got it.
So I think we got our priorities.
Yeah, thank you.
Okay. Anything else?
Yep. Not, yeah.
And then you can work on gallery and other things, it would just
Yeah.
really-
R or anything else. Yeah. Okay.
But
Thank
finish
you.
this today, and tomorrow you can work on the R and helping Grace tomorrow.
Yeah, variation project.
Yeah,
Okay. Got it.
All right. I think
Thank
you can
you.
do it. Yeah.
Thank you. Yeah.
All right. Sungmi. Dr. Sungmi.
Okay. Hi. So I'll do the presentation on a research
Great.
paper Dr. Che asked me to do. I'll share it right now.
So Sungmi, I cannot see your face.
Okay. Yeah. So share screen. Okay. So slideshow. Yep. So, ooh, why is it here? Start from the beginning. Slideshow. So this paper is about so this paper was published in a very prestigious journal, and it was published in July 2018. And it's about comprehensive molecular characterization of clinical responses to PDI1 inhibition in metastatic gastric disease. So I feel I'll make this more clear in the next page. What I found was interesting was like 2018 seemed to be a period where there was a lot of reports on circulating tumor DNA in lung cancer, especially. So I was wondering like what the timing, if it was related anyway. So with the introduction, it was a single center phase 2 trial, which treated 61 unselected patients with metastatic gastric cancer. And they were treated with pembrolizumab. So basically it's a programmed cell death one targeted therapy. So it's an immunotherapy is classified as immunotherapy out of the out of the ways to treat cancer because it's an antibody. And for all these patients, they performed integrated molecular profiling. Basically, by that I mean they performed molecular characterization of tissue. So what would be the goal of the study is to identify the precise predicted biomarkers to select the better select patients with gastric cancer, i.e. those who might benefit most from immunotherapy. So that is just to explain what PDI1 is. That is PDL1. So just really quickly, it means that if tumor cells have this PD programmed cell death ligands, it's a way for the T cell to kill the tumor cell. And by us making this anti thing, we could have tumor cell, we could cause it to die. And also moving on. So with the background, there were other early phase clinical trials, and there were some biomarkers that seemed promising. So these are from previous trials, and one would be, of course, positive immunohistochemical expression of PDL1. And in previous trials, our ORR was 22, meaning like, I'll explain what this is further later, but just to get an idea, it's like the response rate, is overall response rate of the tumor. And the microsatellite instability high versus microsatellite stable. So that's a PDL1 again. And this is what I mean by microsatellite instability. Basically, if there is any break in the DNA, it should be fixed using this mismatch repair complex. But if you have a mutation, then you have the defect DNA that could lead to a mutation, which could possibly lead to series of transformations that could lead to a cancer cell. So if you're, yep.
So the professors
Yeah.
who first discovered this mechanism with repair won the Nobel Prize.
Wow.
And one of them was Northwestern professor.
So proud.
Know that?
No. But now I know. So yeah, so stable is good. Instability is bad. And also one other, so like you can see the big difference here. And also the positivity for the Epstein-Barr virus, EBV, we call it. These are the rationale, like why they think it might help, but I won't go into it. However, they do have some reason why I think it should work. But however, they say pembrolizumab has not been reported as having a response in previous cases. So this is something they want to look in this study. So these could be the potential hallmarks of sensitivity to cancer immunotherapy. And but we have to keep in mind that all these studies were like stage 1 and stuff, so they only had a very few number of patients. Like, so these figures are not maybe that reliable. Okay, but anyways, in other cancer types, there are other options that could be biomarkers that could show promising results in cancer immunotherapy. So in bladder cancer, there's the mesenchymal type, and in lung cancer, there's a ctDNA mutation load. That's what Dr. Chae is doing, so I just highlighted a little bit. And then, so the primary aim of the study is basically what I've been saying before, is to correlate clinical outcomes with molecular and genomics biomarkers to identify patients with gastric cancer who are most likely to benefit from this immunotherapy, specific immunotherapy, pembrolizumab. And to repeat again, it was 61 unselected patients because it's a phase 2 trial, so they have a fewer patients, and they're trying to navigate the trends here that could lead to bigger trials with much more patients with a possible placebo and stuff. But this is a phase 2. There's 61, and patients with metastatic gastric cancer, and then they're treated with, of course, pembrolizumab. And all these data was collected for each and every patient, like tissue biopsy, all these RNA, DNA sequencing, and plasma-derived ctDNA. And baseline characteristics is put here. Nothing too significant. They're all Korean. A lot of them had this therapy as their third line metastatic treatment, and over like 2/3 had more than 2 metastatic sites. I don't know why this is coming all the time. Okay. So this is like a chart which shows, and the results. So with the results, it's pretty interesting because there were 5% of complete response, almost 20% in partial response, 30% has stable disease. So the ORR, which I mentioned before, which is overall response rate, that's CR plus PR, and disease control rate, which is DCR, is almost 60%. That also includes the stable disease. There's not less tumor burden, but at least the size is the same. So that was a result. And the median follow-up was 16 months. So I just put the result first. So overall response rate was 50% in PDL1, almost 90% in MSI instability gastric cancer, and 100% in EBV positive gastric cancer. And we'll go into detail. So response to pembrolizumab is associated with MSI high and EBV positivity. And this is a waterfall plot, which I saw the first time, and it's really helpful. So each and every one of these people in this longitudinal axis are patients, they're numbered. And if you see the color, they're colored like this. And this is on the vertical axis is the maximal tumor reduction. So going up, positive means the tumor increased in size. Going down, negative means the tumor decreased in size. And I suppose this would be like a stable region. And if you see like all the MSIHR and the EBV are at this end, right at the extreme right end, meaning most of them saw a significant reduction. But of course, there's one anomaly here. Not this guy, sorry, this guy, EP48. EP48 had MSI, so microsatellite instability, but for some reason, only 1 of 7 had increase in tumor, and they do go into detail later on. So why does this person have is different? Is an anomaly. So this was a 26-year-old woman with signet ring cell carcinoma, and they discovered through further like progging and that some people, they are actually heterogeneous in their tumor. So she was both she had all these heterogenic cells. Some had instability, some were stable. So, and this was actually a common trend for gastric tumor. I'm not actually convinced just by this, but that was what they suggested. And another result that we want to also talk about is the response to pembrolizumab is associated with PDL1 CPS positivity. So again, those people who are positive, which are depicted in red, are at the right side, meaning there was tissue tumor reduction. And more results coming because this was a study where they saw a lot of factors. Oops.
How do you define CPS?
CPS, it was less than 1, I think, here. Is that what you were asking?
How do you define CPS? CPS.
Oh dear. I'm not sure actually. It's the number of PDL1 staining cells divided by the total number of viable tumor cells. So it's like the percentage of those stained positive.
So those cells could be cancer cells or immune cells.
When it has the C at the front, it includes both tumor and lymphocytes, and by that I mean the whole spectrum of immunocells, because So it's a number of any PD-L1 staining cells we could detect.
What is the cutoff?
I think the cutoff was 1%.
Okay.
Yes, it was greater or equal to 1% in this study.
Okay. You can move on.
Yep. And so like results 4 is, so these are more results. So mutational load and PD-L1 messenger RNA expression were also significantly associated. And EBV gastric cancers, they responded regardless of mutational load status. And also ctDNA, which is something we're working on in lung cancer. It actually predicts response to pembrolizumab, and they use Guardian, which is a commercialized way to get the tumor, any DNA shedded in the blood by tumor cells that you could obtain by doing blood draw. So this is not invasive. So there is something exciting about that. And also change in ctDNA immediately post-treatment does predict response to immunotherapy. So unfortunately, I'm only halfway through this article, which I am very embarrassed about. But so I think I'll have to do a part 2. So
Yeah, so this the dynamic change in Garden ctDNA is the part that I want you to focus because it's very similar to what you're doing.
it is.
And mutational load and is very interesting. And the other thing is, what does EBV do? It's very interesting topic. EBV can cause nasopharyngeal cancer. It's endemic in south part of China. EBV can cause gastric cancer.
Yes.
And when they do, they make the cancer really immunogenic and sometimes cause amplification of both PD-L1 and PD-L2, which is amazing. And who would have guessed that EBV positivity would, 100% predict who's going to respond to immunotherapy. So that's why this, where was this published?
Nature, cancer. Oh, Nature Medicine.
Nature Medicine. Yeah. So very simple. They just combined the clinical trial result. Just looked at all these factors. You can still publish in Nature Medicine.
Yeah.
But you have to have a nice original ideas. So you can we can analyze our cancer patients' data in this way too. Unfortunately, MSI high respond very well to immunotherapy is super rare or almost non-existent with lung cancer.
Oh.
But tumor mutational load is what we're interested and can look at. But from this paper, I want you to learn that wow, people can think outside the box and look at the viral infection as a predictor of immunotherapy response. Can I think something, about lung cancer predictor really outside box? That's number one. Number two, how do they describe the dynamic ctDNA changes associated with immunotherapy response? How did they make tables? How did they make graphs? How did they summarize the results? Can I do similar things with my own data with lung cancer Signatera? Okay, so those are the things that I want you to look at, okay?
Yeah. Can I just comment 2 things really briefly?
Okay.
So number one is I heard that, you know, Helicobacter pylori also causes gastric cancer.
Yeah.
It's a bacteria, and when someone, it was a biochemist, someone who, non-doctor, who first suggested that idea, older doctors were very critical of him, and then he drank it himself and got early stage gastric cancer, like very early stage, and got it treated. And then he got a Nobel Prize for it. So at the time, he was very ostracized by the medical traditional community for his ideas. And I feel like EBV is also something not exactly like that, but it's like you said, out of the box. And number two is I was reading.
He ingested Helicobacter pylori.
Yeah.
Well, he
He-
could get ulcer or gastritis, but it takes
Oh, sorry, he got ulcer, not cancer. He got ulcer, so he showed it. Yeah, sorry about that.
it takes time to develop cancer.
Yeah, sorry, he got ulcer. So, and then that kind of, he did that. Yeah.
Did he
Sorry,
get,
not cancer, but he got ulcer. Yeah.
did he get Nobel Prize for cancer or gastritis?
I have no idea because I only heard it from someone. I didn't read it.
I want you to present the fact, okay?
Oh, you want me to present about it like for a few minutes?
Yeah, next time.
Okay. Yeah.
I don't want you to, oh, I heard this. No.
Oh yeah, I know. But sorry about
I can
that.
tell you a similar story. Listen, it would be Grace. Listen to this amazing story. This is Dr. Harrington or Hamilton. I think it's Dr. Harrington. I'll look it up. But so, do you know ITP, idiopathic or immune thrombocytopenic purpura? It's a disease where your platelet drops, and you start to have purpura, which is like bruises or rash. And people didn't know why people get these disease. Now, this hematologist discovered that it's the autoimmune antibody to platelet. And he wanted to show that it works, that it is the mechanism. So he got the antibody from patient's blood, he injected it into himself and showed that his platelet dropped. And
Wow.
he didn't want to sleep with, he slept upright because he didn't want to have hemorrhage in the brain. But he showed that the antibody was the cause of platelet drop in human. But he survived because he got, I think he got steroid, and it increased the platelet count. So what a crazy hematologist. He didn't get Nobel Prize for this, but yeah, you get to a level to prove anything. People get to a level where they can sacrifice their health, and it's crazy. But for scientific belief, they would do it. You know, think about this. Yeah. But anyway, you go on.
So thank God we're not in a position we have to do that. But research is still fun. And my second point was that the reason I thought this would be in nature is because how it was very comprehensive. It didn't look at a few things. It looked at a lot of factors at once. And it was a very well written piece, and it used all these great tools to present their information using very diverse way to show them, like the box plots and like that waterfall plot, and I just was impressed by all the things they used. Yeah, and so I thought like, oh, this is an example of a very well written article. So yeah, it was just I actually had quite fun starting to read it. So I only started reading yesterday, that's why I'm not finished, but yeah.
Not difficult. You can do it too. This is just a very simple correlation study. Yes or no, and was there a response or no response? There's no deep science, there's no deep calculation into this. This is like, how do you make it look pretty? How do you present the data? So and then do you have the idea? Do you know the field? Someone talked about EBV. Okay,
Yeah.
they're not the first group to say EBV is linked with immunotherapy. They're not, but they were able to know the trend and apply that into their patient database.
So they would have got this information here in red from someone who's doing basic science about and then they try to put it into clinical trial. So this part here.
Pembrolizumab response has not been reported in EBV positive cancer patients, but probably nivolumab has one. Probably other immunotherapy has one. So all these references were there, so they knew there was a clue. You know what I mean?
Yeah.
Then you can look at, well, in my specific group, this one was never looked at, so I'll look at it. You know, so science is finding a niche. Who found what? And what data do I have? And how can I fill that gap?
Yeah.
That's science. That's called translational science. Bridging what's discovered. So you have to be up to date. What is being discovered? You know, what's the science cell paper? Now I want to bring that to my clinic. And what data can I gather from my clinic that can apply the principles found from the lab? So that's what we're trying to do. So, you know, chromosome instability, how do you calculate that? You know, mesenchymal signature, how do you calculate that? So study and present the later half of the article next week, okay?
Okay.
Okay,
Yeah.
what about your project? Do you have any update?
Yeah, I did fill up like the rest of the, but I didn't analyze it yet. So I feel like yeah, I have a deadline now, which is this Friday. So I work better with deadlines. I'll just try working on it. I have to just do it from now. Like, I did put all the data in there, but yeah, I will have to search for some kind of something new, something that's
So if you find some new trend, that's good. If you don't find it,
yeah.
just report the landscape. How many
Okay.
students had positive results? And you know, some people have repeated 4 times and it was still negative.
Yeah.
You know, you can make a table. How many times was it repeated over what time, and was it all negative? And did anyone turn positive? Did anyone was positive even from the start? You know, what was the percentage? And when they were positive, we asked, so did Natera email us back? Did they say, can they
I haven't sent the email, sorry, because I wasn't sure what to send it, but I'll send it today. I wasn't sure what to send the email, so I didn't send it.
So I told you what to send it. So if you don't understand, you have to ask me, and you have to give them a week notice in advance. The deadline's this Friday. I don't think it will give us anything by the end of Friday.
Okay.
So this is the very nature of collaboration. Like if you're not the only researcher, you have to, you know, respect their time.
Yep.
And any like communication that happens has to happen in a timely manner. Otherwise, we won't have quality data. Okay. So don't assume anything. Don't assume that they will get back to you the same day. So the question whether we do the abstract or not, I mean, the question we're asking is everyone gets 16 gene personalized gene panel. We want to know what those 16 genes are. It's not reported in the panel. Okay. You can guess what they are from their Altera, which is the tissue-based NGS report.
Yeah.
but sometimes it's not clear. Some of them we don't have Altera.
Mhm.
So we want to ask the company, can we get 16 gene list from everybody?
So everybody has it. Everybody's is personalized based on their Altura. They get 16, and then that's used for to do Signatura.
Yes.
Oh, I get it now. Sorry. And I'll send it right away.
That was what was written in my JTO review paper. That's why it's called personalized ctDNA panel versus Guardant, which is
Not yeah.
just a set panel for all tumors.
Yeah.
So the question is, what are the most common gene mutations in lung cancer
Mhm.
are used in personalized gene set? You can you can show them the frequency and what seems to have the highest allele frequency. So not only the 16 gene and its individual variants, but also the VAF, variant allele frequencies that are measured in Altera, which is the tissue NGS, not the blood NGS.
Yes. So
Um,
like we could do like this paper looked at each factor. We could also maybe do that after we hear back from them. Like for
so
each of the
if they
more
get
common.
back to you in a timely manner, you can do the analysis that I just told you. If they don't, you can still describe the landscape I just told you.
Yeah.
Still write the abstract.
Okay.
Okay.
Yep.
And then PDL1 positivity, any, the EGFR mutation, any association you want to see with the variables that you've already collected, and if you can find any association, new trend, you should report it.
Yeah. Okay.
Questions?
Not right now. I think I got what all that you said. I think I understood.
Okay. Anything else?
No, I tried writing an abstract without any data, so I just realized writing the abstract is not the problem. It's this part. It's like the questioning, finding the that way of thinking in transitional research. So yeah, I have to do it actually. Okay, thank you so much.
No problem. And if you don't know, feel free to ask.
Mhm.
And you can also ask Dr. Eugene O
Yeah.
or Iryong Chung. Anyone. You can talk to Minjong, Kim. You can ask for help and they can help you.
Okay.
I think the greatest joy of doing scientific research comes from discussion. Like I'm kind of stuck here. What would you do? Like looking at this, can you find any trend? Can you give your input? That's how you learn. Some people have amazing insight. So my hobby is always, can I pick your brain? And then I and I throw a question at them. Can I pick your brain? Like, can I show you this graph? What do you think about it? So make that your habit. That's the only way you're gonna get better. You don't wanna just think and then, oh, I can't make any progress. The reason I go to all these scientific meetings is that you hear from the world's, guru on this topic, but then you can, after the talk is done, you walk out and then ask them question. Hey, I have a really good question for you. Like, I wanted to ask you this question. And then you just pick can I pick your brain? And then you just tell them all these questions. They'll tell you. They're amazing. Because all they do is think about that, similar question. You know what? That's why I fly there to talk to them. I can listen to their lectures from YouTube and everything, right? But so make them your friend. Next time you write a paper, invite them as a co-author. That's how you become friends. That's how you can continue to pick their brain. Okay.
Okay. That's
So
amazing advice. Thank you.
so yeah, I want you to learn the joy of collaboration. It's painful to get things done, but idea coming up with asking good question is 50%. And then, you know, getting to the breakthroughs, leading to good collaboration, you know, this is what makes us going. And finally, the outcome should help our patients. Like, I'm not doing this to pat my CV or my promotion. You know, I'd rather go on a vacation than do anything to do that. I do it for the sake of my patients. Okay, so there's urgency to that because this is clinic. All right.
Just one thing to add. Yeah, I feel like it's also there's a race. But Sally said she wants to be involved in any research in the currently going in the lab because she actually wants to discover more about research and like be more involved and be understanding. So I said she could definitely help in my research. And she just wants to say like it's not about her name being on it. She wants to do more than as much as she could to learn about the world of research. So I'm just letting you know that she will be a collaborator.
So I know Sally has class this time, but you know,
Yeah.
Eileen, whoever. So this is the rule. Whoever comes to our lab in person or to our lab meeting through Zoom, they can reach out to the lead investigator, which is you for this project, and work with you in whatever capacity they can. And you are going to be the one who's going to tell me they've done this, because I can't supervise, you
I
know.
know. I have a friend who did PhD in the MD PhD in DNA, but okay, I'll talk about this later, in person.
Yeah, you can work with any undergrad who's willing to work with you. So that's kind of the rule.
Okay.
Any question
Nope.
before we wrap up? Okay. All right, Grace, keep me informed. We got plans for each day. I think we can do it.
Yeah,
All right.
I've been working on some things right now, but I will let you know.
Okay. Super. Take care. So you have to stop the recording first.
Yeah.
then-